Yes, so thanks very much, Jamie, and welcome everyone from my side. My name's Alannah Brett, and I'm joining you from Switzerland today. And I'm very happy to welcome our speaker, Richard Herrington. So thanks, Richard, for joining us today. And I'll just give you a short intro. So Richard is the head of Earth Science and a merit researcher at the Natural History Museum in the UK. And he has quite a legacy at the NHM, having first been a researcher there in 1991. He also holds a visiting professorship at Imperial College. And Richard's main interests lie in investigating the behavior of metals critical for our modern economy in Earth systems. And he specifically focuses on metals concentrated in deep weathering processes in the Earth's surface systems. And his projects focus on looking at cobalt and rare earth metals in particular. His work involves a lot of collaboration with industry, and as a result, his research can provide new information enabling better and more sustainable mining techniques. His other research interests include investigating links between mineral deposits formations and the Earth's geodynamic history, and the association of mineral deposit formation and life in the deep ocean. Now, Richard's collaborative work with those in different fields sets a great example for what can be achieved in interdisciplinary study, and that includes his research, which he's talking about today. So Richard, really looking forward to your talk, and Jamie and I have gone on long enough, so I'll pass it over to you, and we look forward to hearing about the connections and insights between metals and early life at seafloor vents.
Brilliant. Well, thanks very much, Jamie and Alannah, for the kind introduction. And yeah, just thanks for the opportunity to speak. And hopefully I'm last but no means least. But good to talk to everybody. Yeah, today I'm going to talk about something that is a little bit further away from mineral deposits, perhaps, than some of the talks you've heard earlier. And it is really looking at the very interesting relationship between unusual life forms in the deep ocean and the ore deposit formation. But of course, that may have implications for origins of life. As you go through the talk, you'll probably get that gist. Like all good things, there's always a team involved. And although I'm giving the talk, I really want to thank the team that I've been working with for the last 4 years on this NERC project, particularly Maggie Georgieva, who was our postdoc, but the team, including Adrian Glover, Chris Little, Anna Riesgo, John Copley, and Valery Maslenikov, who I've worked with since the early 90s, when Valery was one of the few people that recognized the presence of fossil material in massive sulfide deposits in the Urals. And he and I made a connection that started a project that has been literally running and running. Chris Little was our first postdoc. Now Chris is the world expert really in this field, and he's based at Leeds University. And I just really want to give thanks to the team. We've worked together on this for the last 4 years. I'm going to take you on a bit of a dive to start with. So hopefully this will work. So you're in Bob Ballard's ROV at the moment, and you're somewhere off Galapagos Islands. And you're an exploration geologist, and you're excited because you've just found what you're looking for. And that's an ore deposit forming on the ocean floor. So what you see there is obviously the massive sulfides building as chimneys. You've got the hydrothermal fluid venting and forming minerals in front of your very eyes. But then as you zoom in, you start to see that there are interesting colors there that are not just minerals. There's a whole lot of white on the edge of those chimneys. And as we zoom in, you start to see there's life there. There are these really weird crabs living there. And they're now just going to zoom in a little bit further up into this area of white, which is all organic material. And you'll start to see some little red, look like little palm trees. And these are actually known as palm worms, and these are alvanellid worms that are living right on the vent chimneys. Very, very unusual life forms. So this was discovered in like late 1970s, and big surprise to everybody that there are all this life there because it's such toxic environments. And this is really what spurred me to start to work on these and think, well, these things get preserved in the fossil record. Is there any evidence for these kind of weird animals in the fossil record? So where do we start? Well, we start with looking at location of modern high temperature vent sites, because that will give us a clue as to where they form in geological environments. And you can see from this diagram here that they occur at a range of plate boundaries. The obvious ones are the sort of mid-ocean ridge spreading zones, divergent plate boundaries, but we also find them associated with convergent plate boundaries, but in extensional sites in convergent plate boundaries. Because in order to get these things going on the seafloor, we need extension because there's a big involvement of seawater. So moving that on, some nice published diagrams, which I've kindly borrowed for the talk. I know you've had some talks on VMS systems, so this is probably a little bit of a 101 for most of you. But I just wanted to remind you that we're looking at areas that need crustal extension, the circulation of seawater, and the interaction with magmatic systems. And it may just be as a passive source of heat to heat up the seawater and drive the hydrothermal systems. But we do know that in some of these cases, there is an implication of a direct magmatic fluid into the ore bodies that form. So basically, the little cartoon on the right shows you what happens. Seawater gets driven through close or adjacent to hot rocks, in this case, the magma chamber at the mid-ocean ridge, and those fluids get expelled onto the seafloor after having dissolved a whole bunch of metals from the footwall sequences. So you all have seen this cartoon, no doubt, as undergraduates or a graduate school, of what sits underneath one of these seafloor hydrothermal vents where sulfides are forming. And this is the classic diagram Mark Herrington and colleagues put together after drilling the TAG mound. So that's the transatlantic geotraverse massive sulfide that was found and drilled in the 1990s. And this classic diagram shows you the morphology, if you like, the characteristics of what one of these systems looks like. And what we have, obviously, there's a lot of things happening below the seafloor. But what I'm really interested in, what I'm going to be talking about today, is what actually happens at the seafloor itself, where we get the top of the massive sulfide lens. We have hydrothermal fluid that manages to make it break through onto the seafloor. And you have the formation of those black smokers that we just saw in the little video clip. But we have a whole range of other environments. We have sediments forming. We have chemical sediments, but we also have clastic sediments. The chimneys break, fall down. And then, of course, we have those animals that are living in that active environment. And the animals are pretty much linked in to the venting, but also the fact that you've got sulfide minerals, and it's sulfur's the key. A lot of these organisms, the life is based on chemosynthetic bacteria that are reliant on the sulfur from the venting, and we're going to be exploring that as we go onto the seafloor. So I just want you to remind you of those environments that you are going to find on the seafloor. And it's thinking about what those environments would look like in the fossil record, because that's where those fossils could get preserved, the fossils of the animals that are living around the vent sites. So there's just a cross section. We do know of some of these. We've drilled some of the other sites. So that the tag was right at the mid-ocean ridge, a spreading zone. But this one here, which is Solwara off Papua New Guinea, is in a back arc setting where, again, you've got extension. It's a super subduction setting. but we have the same system developing. So you have an altered stockwork feeder zone, and you have a massive sulfide lens, but you also have these venting chimneys on the surface, and probably all very aware of the work that Nautilus Minerals did in defining a seafloor resource of sulfides. And it stands as the best sort of natural laboratory to understand these processes. We also know that these massive sulfides don't only just sit on the floor, seafloor, we find them forming subsea floor replacements. And in the geological record, a lot of the sulfides that we find could well be the process of seafloor replacement, subsea replacement, like we see in this case, the Middle Valley deposit in the northern Juan de Fuca. So it's not only a simple seafloor story. So when we are going back in the geological record to look for evidence of these animals that might be living on these sites, some of the massive sulfides might be completely barren because they formed intraformationally by replacement. So quite a lot of the deposits form in that way. We're not likely to find any animals associated with them. But on the ones that are forming on the seafloor, we hopefully would have a good chance that organisms would have been there if indeed those communities were alive at the time, and they then might get preserved in the fossil record. So actually, when we're going to look for things in the geological record, we should be thinking about the kind of textures that tell us what that we were really on the seafloor. And one of the most diagnostic textures that we would have would be the black smoker chimneys themselves. These are the things that form on the top of the mounds. And you can see on the diagram on the left-hand side there, that's Meg Tyvee's cartoon, really nice cartoon, that these chimneys form with a high temperature fluid venting up through the center of an open conduit. And you get zoned mineral development around those conduits. And they're very, very distinctive when you find fragments of those on the seafloor. The picture on the right there is a fragment that Rachel Heyman had brought up from one of her dives. And you can see what you've got is this is a cross section from a chimney fragment. You've got an open conduit in the middle that's formed, it's got sparry minerals, but you've also got that sort of golden ring of chalcopyrite that's forming in the high temperature zone. So that represents the very high temperature part of the system. And then as you go further out, you get pyrite, sphalerite precipitating in the cooler parts of the chimney where you get diffusive flow through the edge of the chimney. And so this is a classic kind of texture, and it can't be interpreted in many other ways. So it's one of those diagnostic textures we look for in looking at fossil VMS systems. All right, so what are the ecosystems on the modern seafloor? So you've probably seen these pictures of the spectacular vestimentiferan tube worms, and these are, I mean, they're quite beautiful animals. They're oxygen breathing, they're red because they've got a hemoglobin, so they live right on the seafloor. So we know that these things live in oxygenated seawater, and they're often associated with a whole range of other organisms there. So we've got vent clams, we've got barnacles living on the clams, and we've got juvenile gastropods that are climbing over everything and feeding. We've got little fish. And on the right there, that's a sort of a bit of a trophic structure of one of these vent sites where you have a whole range of organisms that are living in and around the vent. And so the question is, where are they getting all of this? Where are they getting their food from? They're in a pretty barren, deep part of the ocean. Well, the answer is there's a lot of chemical energy there, and we've got a lot of bacteria living around those vents. So photograph on the left-hand side shows the white there is actually bacterial flock that's basically forming around these vents, and they're feeding off reduced sulfur. So they're using the reduced sulfur to sulfate transition, the energy from that, they can use that as an analogy really of sunlight. So that's chemosynthesis rather than photosynthesis. And so then that bacteria becomes the basis of the food chain. And some of the animals have been really clever, like the tube worms. They actually don't have a gut anymore. They basically fill their inside of their body, where their digestive system would be, It's full of symbiotic bacteria that are utilizing that sulfur, and then they're producing the organic molecules that the tube worm lives on. So the tube worm's being very lazy. It just gives a free ride to the bacteria, feed it from the sulfide vents, and then it very cleverly avoids getting poisoned by hydrogen sulfide and has a very good life based around the chemosynthetic bacteria. And then some of the vent mussels on the right there, that's just a cartoon to illustrate that the vent mussels have developed bacteria that live inside, again, symbionts in their gills, which are able to use actually hydrogen, and they are effectively organic fuel cells. They're using energy from the hydrogen, the acid coming from those vents is actually then being used by bacteria as a fuel. So their fuel, then the mussel gets a benefit from the life cycle of the bacteria. Okay, so there's an intricate link between why the organisms are living at those vents and the actual sulfide formation. And actually in their life, there's quite a close link too. So a lot of those animals that live there are building tubes. The worms build tubes, and partly that is to protect them from hot water. These little alvanellids, and we've got a picture there of an alvanellid tube on the left-hand side, and the palm worm that's coming out of the tube. But that tube actually protects them from hot water. But these things can live up to about 90 degrees Celsius. They're the most heat-tolerant multicellular organism we know. But actually the tube is mineralizing while the animal's still alive. And so the animal is fighting a battle against the growth of sulfide. And eventually the sulfides take over. And on the right-hand side there, we've got a bit of, this is a vent tube that's completely replaced by pyrite. Actually, as the animal is, shortly after the animal leaves the dwelling, it dies, the tube empties, and then you get the sulfide overtaking the whole of that organic matter. So really nice piece of work our student did back in the early 2000s, Ellis McGinn, looking at how that mineralization process proceeds. And then what you leave, this is some images of sulfides that have been pulled off the seafloor from the East Pacific Rise. And you can see, though, that's a complete replacement of those organic textures by sulfides. And you get delicate preservation of the layered tube wall. And on the right there, you get the ghosts of even the bacteria that were growing on the margins. So if I go back to the picture here, you can see in the middle photograph there, there's a whole lot of stuff dangling off the tube wall there. And these are filaments, bacterial filaments that are growing on the tube in its life. And these are processing reduced sulfide and produce native sulfides, and then the sulfides that the tube get replaced by. So there's a straight link there between the organisms and the growth of sulfides. And you can see these things are replaced in sulfides. So we've got a really good opportunity that if that sulfide gets preserved, we might be able to go and find that in fossil systems, that we've got evidence that there was life there before. So we've got a lot of documentation from modern vent sites. A lot of organisms have been studied and looked at, and now what we were proposing to do was to go back into the geological record and track those organisms there. So there's just some photographs of photomicrographs of how pyrite has preserved organic structures in material at modern vent sites. So these are from East Pacific Rise. And you can see on the right-hand image, beautiful, beautiful ghosting of the filamentous microbes within the tube wall of some of these alvanellids. So the big question was, can we find these fossils preserved in ancient sites where there's perhaps been low metamorphic grade? So of course, the first thing we did in this was to look at the geographic distribution of the MS deposits and the geological time distribution. So this is a compilation from Jim Franklin's 100th anniversary paper in the Economic Geology, which documents. We had a whole range of places to go, right from the Archean all the way through to the Cenozoic. We've got VMS deposits forming. We know VMS deposits form when you make oceans and when you close oceans. And when there's extension in submarine environments, in high heat flow, you're going to get these hydrothermal systems that could potentially form sulfide deposits. So effectively, huge number of belts, up to, well, we've got 42 here to target to go and look for those. So how did we start? Well, this is where this vent project came. So what I'm going to be talking about tonight is based around this paper, which you can download. It's open access in Earth Science Reviews. And if you're interested, please go and have a look. More of the details in there. But this vents project started on the back of a project that we had in the early 1990s. I said we started work in 1995. And Chris Little, who was working with me then, put together a compilation of what was known then. And we had 19 recorded fossiliferous sites when Chris wrote this paper, which is in a special edition of the JOSOC special publication in 1998. And what we've done with our vents project now is we've extended that to 49 sites, and we think we may have 50. We've got another one we're looking at the moment. 49 sites now documented. And we've expanded, obviously, by a factor of 3, the number of sites. There's still not many, but what we have done is systematically target particular bits of the Earth's, these VMS belts to look for those. The next diagram really is just to give you a summary before we get on to looking at these things in a bit more detail. Where are we finding those in the geological record? And you can see we got them all the way stretching from the Cambrian all the way through, still call it the Tertiary up here. Obviously it's the Cenozoic, but you can see there are big gaps in our knowledge, but we've got some big blips, and they more or less coincide where we've got large development of VMS deposits. So the first place I want to go to is actually the oldest of the camps in the 1990s that we looked at. And this is where my colleague Valery Maslenikov came in, because he'd recognized fossils in some of the massive sulfides there. So we started looking systematically in the Urals, and we've now come up with 25 deposits that they're shown on this map here, which is in our paper, where we found these massive sulfide with fossil communities preserved. And this is testament to the fact that the Urals is an incredible, the Silurian to Devonian sequences in the Urals are fantastically preserved, really very, very pristine. And so a range of settings, you'll see the paper on the tectonic settings on the right, all extensional systems related to arc volcanism. I'm just going to show you 2 examples, Yemen KC, which is the Silurian example, and then Subai, which is Devonian. So the Yemen KC deposit is quite small. It's a small, massive sulfide, very well formed. Here's a little map and cross section. You can see it forms a sort of a classic lens, tilted lens in the open pit that's developed on a stockwork alteration zone. And so what we did there, because we had such a small, cute little deposit, we could work on it in toto. We took the whole thing apart and were able to link together the ore facies that link to the ocean floor, and through that we were able to discover the presence of fauna. So one of the key things that we did identify was evidence of black smoker chimneys. And these are beautifully preserved. Top right there, you can see that's a cross-section of a conduit with that classic chalcopyrite ring. We then have on the left-hand side here, talus breccia, and you can see in there chimney fragments, beautifully zoned with chalcopyrite rings in fragments of vent chimney. We've got evidence of mixed sulfide and oxide sediments, tells us we're on the seafloor, oxygenated seafloor. And then we had these wonderfully preserved vent fossils. And you can see that slab in the middle there, that's totally massive sulfide that's totally preserved in, which is preserving the casts of fossil material. So there is no shell left. So there are any carbonate that was part of the shell or any organic matter that was the tube is no longer there. It's all completely replaced by sulfide. But you can see beautifully the textures are preserved, and we are able to recognize these as organisms. So in the case of Yemen KC, this is just over 950 specimens of fossils were recovered from this site. It's not just an anomaly, one or two pieces. And we were able to see that there's a complete range of organisms from what appear to be worm tubes, so potentially tube worms living there, all the way through to these vitagastropods, monoprocophorum, which is a very early mollusk type, and then these large tubes and a very large brachiopod. That's a 10mm scale, very large. So hugely diverse from that point of view. It's not just one organism. We've got filter feeders, we've got the worms, and then we've got grazers like the mollusks. And actually, one of the really neat things from my point of view is to find fragments of vent chimneys on the surface of which we were able to find the thin tubes. So just like we find today that modern alvanellid sitting on a modern chimney, we found these dwelling tubes on Silurian vent chimneys, so 420 million year old chimneys. And not just a small deposit. So this is the Subai deposit, which is in the Devonian. It's a huge deposit. It's more than 100 million ton massive sulfide. So it's not just these small deposits that have this venting and so a lot of big deposits probably form by replacement, subsea floor replacement. But in the case of Subai, we have clear evidence that this formed on the sea floor. So it was a 100 million ton massive sulfide lens sitting there on the sea floor. So get your head around that in the geological process field. It's quite stunning. And Subai also had less diverse fossils recovered there, but over 130 specimens. Some of these vestimentiferans, whether we can call them vestimentiferans, we're not really sure. They're certainly dwelling tubes that morphologically resemble these tube animals, these tubular dwelling animals. But on the left, we do have a bivalve, which is definitely a bivalve and would have been a filter feeder sitting. Yes, so just really to show, and the one at the top's also pretty interesting, H there, where there is a scale. So this is actually from the Subai deposit, that giant massive sulfide, where we have a good preservation of, you can see this open conduit, and then the tubes are right on the periphery of this. So this is clearly a fragment of the chimney with the tubes that are being preserved on the edge. So we've got no doubt really that these organisms were living there, performing the same function as they do at modern vent sites. And so I'm just going to take you on a very quick trip now through the rest of the geological record and give you a few snapshots. So one of the deposits that we looked at was quite a small massive sulfide in California called Figueroa, brought to our attention by Rachel Heyman. And it's in the, basically, it's part of the accreted terrain on the west coast of the US, but it's a Jurassic, probably a primitive arc that got accreted on the western side of the Americas. And again, we found quite a diverse assemblage of fossils that represent a community that were living around the vent site. And indeed, we found one small fragment. This is a millimetric scale. It's quite a small conduit, but I think you'll be fairly convinced that that type of texture in chalcopyrite would be unusual to form by any other means than being a fluid escape conduit that would be connected to the seafloor. And so there was good evidence here, again, that there's, in this case, we've got tubes and we've got gastropods. We've lost the brachiopods in this case, but gastropods are there. And then in the cherts overlying the VMS deposit, we've got good evidence of filamentous bacteria. So this is actually in the chert that's sitting right above the massive sulfide, and you can see even evidence of the internal cellular contents of the filaments. So these are probably the bacteria that were thriving at the vent site that gave then the basis of the food chain for all of the animals living there. And then in the Mesozoic, also the Tethys has been quite productive for looking for these types of deposits. So we've now been looking at 5 different sites in the Tethys region. So there's a Troodos ophiolite itself on Cyprus. There's Samael, which is in Oman, and Rachel Heyman and Randy Koski worked on this and found some evidence of fossils there that we were able to look at. But then there've been some new discoveries, Chaeli and Alannah in Turkey, and then Madnuli in Georgia. And so these are actually arc-related, back arc systems. So the Troodos and Samael are probably 4 arc, primitive arc systems, but definitely the ones in Turkey and Georgia are in evolved arc. So we know that in the Tethyan arc, we had similar kind of communities. So Troodos has been remarkably productive. So 6 of the deposits, 6 different sites have been discovered there. Again, a range of organisms from tubes, vestimentiferan tubes. And in this case, these are typically curled tubes. And we know that in the modern ocean, we do have curled vestimentiferan tubes. So this is why we can be a bit more certain about these being vestimentiferans, not just simple dwelling tubes. And then again, you see the invasion of gastropods into the Mesozoic sequences. And so these are all perfectly preserved in sulfide. There is no shell material left. These are just perfect casts of what the shell, where the shell was. And then just the Madnuli deposit in Georgia, we just have tube worms or tubes that were probably the dwelling tubes for vestimentiferans, we're guessing. And then the Chaeli deposit in Turkey. Also, these are actually in drill cores. We've not found any in outcrop, so we can't be able to describe these very well, but you can spot them in drill core if you're a smart geo. Actually, the geo logged it didn't, but Valery Maslenikov did, because he recognized that these are important, biological structures. And then in the Cenozoic, we also start to find some too. This is actually from Claveria in the Philippines, where this is associated with volcanic tuffs and venting. So all the way through the geological record, good evidence of these organisms. So I'm going to get to the kind of denouement now of talking about what are the conclusions from all of this. I think you've got the message that we were successful in tracking back these vent communities back into the geological record, and we think we've got them back as far as the Cambrian. And so now my biology colleagues were looking at the relevance of that. What are they telling us about how life has either started or evolved at vent sites? So previous work actually had suggested that the organisms at modern vent sites had resulted from Cenozoic radiation. So they weren't very old. And actually our work has confirmed that to some degree, but it's pushed them back. Our work has shown that quite a few of the organisms that we see at modern vent sites were probably there in the Mesozoic too. And so we've pushed back that time anyway. Vestimentiferans have also gone back into the Mesozoic. Now that's quite interesting because the genomic models would have them only radiating in the Cenozoic, but that could be wrong. Well, it must be, because we've got evidence that the fossils are much older. The problem we have is going back into the Paleozoic. So we've got organisms down here in the Paleozoic, but we can't directly link those to organisms that live today. And despite morphological similarities, we can't link directly lineages back then to now. Even though we see organisms that probably lived in the same way, in the same site in a vent, we can't link those because these organisms are identified on soft tissue. And as good as the preservation is, we don't have much in the way of soft body tissue. Well, we have nothing where body tissue preserved. So taking that interpretation further might be more tricky. So I'm just going to give you the final sort of perspectives on this. And you know, well, why is it relevant to geology? Well, we know hydrothermal vents have been important habitats for life, perhaps from the very beginning. Multicellular life probably been there since the Cambrian. And now, did life begin there? It's still a debate, but Dodd et al. In 2017 found evidence for microbial life basically as soon as mineral deposits started forming. And there's evidence that the last common ancestor, Luca, was thermophilic and lived in sulfur-rich environments. So kind of that's what these vents are. And we've got biosignatures, not buoy signatures, biosignatures reported from a range of Paleo-Mesoarchean vent environments. So probably bacteria were there right from the get-go. And we're doing some work, interesting work with Steve Piercy and his team on some deposits in the Cambrian and the Tally Pond, where we think metazoans might well have started to move into vent sites in the Cambrian. And that's something in prep, and hopefully something will be coming out soon on that. But we know in the Urals and the Paleozoic that faunas were exploiting vent niches effectively as far back as the Silurian and probably Ordovician, likely with symbiosis and bacteria. And we've found vents, fossiliferous VMS, that relate to the abundance of VMS in the geological record. And so we think all eras should be equally prospective, but exceptional preservation is needed. There's possibilities. We don't have any Triassic VMS to test this hypothesis, but there may be some big changes at the end of the Permian when there's a big extinction event. That needs to be tested. But the bottom line is new sites are sought. So keep your eyes open. The reason that we've had success here, we've got some people with clever eyesight like Valery Maslenikov, Chris Little. I have found a few in my time as well. I found the vent chimney material in the Silurian in Yemen KC. I don't think people have been looking for it. So I would say to you all out there, please go and keep looking. And for particular interest for us would be to work on deposits that span the Mesozoic to Cenozoic boundary. And this is where the big changes might have occurred. And looking at extinct and recently emerged SMS, seafloor massive sulfides could be a good target in this regard. Okay, so a little bit longer than 30 minutes, and I apologize, but thanks very much for listening to an extended talk, and I'm more than happy to take any questions.
Thanks so much, Richard. That was really fascinating, and coming from this perspective of mainly focusing on the geological side, it was so interesting to hear the biological side, and the specimens are really amazing. I've never considered fossils completely made of sulfides before, so it's really interesting. You're very welcome to anyone have any questions, write them in the chat. I maybe have one to start off with, actually, and that is, Richard, in terms of like reflecting on the geology of the vents, is there anything that certain organisms being present can tell us about the fluids that were present more than we already know?
That's a really good question, and it's something that my colleague Valery Maslenikov has some very strong views on, because obviously the nature of the vent fluid as it emerges dictates whether you've got things like reduced sulfur, what the availabilities of other trace elements in there. Overly toxic environments might be detrimental. So it's kind of early days, but Valery has got some ideas there that we could actually, some environments, he said, you'll never find vent fossils there because particularly in some of the, what he says are white smoker environments, certain organisms wouldn't be there because there's probably not a lot of reduced sulfur there. So if you're just getting sulfate, you may well not have organisms. So I think there is some evidence that could be used. And at this stage, we probably just don't have enough data to be able to publish and say that. But I think it's something very intriguing, because we do know in the modern sites that particular organisms favor particular types of vents. The tube worms seem to just love it. If you've got reduced sulfur where they can put some bacteria they can have this symbiosis with bacteria, then they seem to be ubiquitous. But there are sites like the mid-ocean Atlantic where there aren't any tube worms, and there is some discussion why aren't they there? And maybe it's something to do with the chemistry, but it might be just the biological distribution of them.
Yeah, super interesting. Well, it seems like there's going to be some more papers on this in the years to come.
Yep, definitely.
That's really great. Okay, we have another question in the chat. Jamie, do you want to read that one out?
Yeah, sure. So it says here that, thanks for the talk. Some of the elements are biophiles, so can we use the finding of your research to explain the elemental fixation via bioactivities and the size of the deposit? So I think this actually is a question I was going to sort of ask as well, in the sense that does the organic do the organisms and these sort of organic processes enhance in a way the elemental fixation and potentially the size of any deposits?
Yeah, I mean that's a good question. I mean, we don't have evidence that we have. Well, the best, the most diverse community we found is at quite a small VMS deposit, so Yemen KC, and what you've got to remember is that the fauna is only going to be preserved on the snapshot of the very last bit of venting on the ocean floor, because we know through zone refining, these mounds grow and they're going to overprint the fossils. So I think the big problem is in those big systems, there probably were a lot of organisms there that could have helped in some way. And I've no doubt that the bacteria are helping fix some of these, particularly the sulfur. They're involved in the sulfur cycle, but they would be just overprinted. So I don't think we've got a view. That's something where we could look at modern sites to, and I don't see much literature on that as to the linkage between the size of modern seafloor systems and the presence of organisms. So there's a great research project for someone to do in the modern environment, and then we could perhaps extrapolate it back once we've got that sorted. Use the modern laboratory.
No, that makes sense. So as it goes, you mentioned about being overprinted and things like that. I think it's clear, certainly from the amount that we found, that there is certainly a preservation bias in the record with these things.
Yeah.
So, do you find that the vent fauna sites that you found show less evidence of remobilization?
Yeah, I think that's pretty true. That the Urals is a classic example where the vent chimney material we find at Yemen KC still preserves isocubanite ex-solution lamellae. And we know that if you heated that up to 200 again, it would be obliterated. So these rocks have not been overprinted. They're, prenite, pumpelliite fascies, metamorphism. So as soon as they get overprinted, you'd lose a lot of those textures. So, one thing that happened in the Urals, it was a very short, very dynamic ocean, but only 10 million years it was really productive for a lot of those big VMS deposits, and it shut off really quickly without overprinting. So I think that's partly the preservation story, is that it's almost like a system that there was something dramatic happened and everything stopped, and then it got preserved, and I think it's some kind of catastrophic termination to hydrothermal activity. Because if it just kept going, a lot of VMS systems have hydrothermal activity in the hanging wall, which suggests they get covered and then there's still kind of activity. Those are systems you're never going to find vent material, fossil material, I'm sure.
That's good sort of to know.
Maybe can I ask a quick follow-up there? So would that also be the case in like a diffuse venting zone that you also wouldn't expect? Or would you still maybe think that it could exist?
You do. Because you get bacteria in some of those diffuse flow zones, but the volume of the fluid is an issue in those cases. But you get thick bacterial mats. You'll have organisms coming in a way they're the secondary because they would be grazing, like those crabs. They probably don't have symbionts, but they're eating the dead animals. So they're eating the dead tube worms and so on. They're scavengers on those organisms that have died. The gastropods, we know, are eating the bacteria. You know, they're grazing them as they would algae or whatever if they were in the photic zones. So, so I think in diffuse flow areas you can get those, but they would probably be some of those larger, people, organisms that were coming in from the outside to,
rather than living there,
rather than being, definitively based on the mineral energy coming from the vent.
Okay, interesting. And in terms of like temperatures, is there any vent that's too hot, or is it like kind of mixed with seawater by the time it comes out where the animals are that it's okay?
Yeah, I mean, the ones that live on the chimneys are playing a dangerous game because literally between a rock and a hard place. I mean, the seawater there is about 4 degrees. The vent fluid's more than 350. They've got to find a place that they can live where they can get the reduced sulfur, so presumably the sulfide plume or dissolved reduced sulfur, but not be too hot. Because I mean, the most tolerant of the metazoans is the alvanellid, which is 97 Celsius. Even they boil, but they can survive that. The bacteria we know can survive higher temperatures, but even them, about 120, 130 bacteria croak. So it's a difficult situation, difficult environment. They live right on the edge. There's such a lot of gain by living close to it that they almost like they take the risk of living. So they build these tubes on the chimneys, but they quite often will pop out of that and build another tube because theirs might get too hot.
Ah, interesting. Okay. Huh. So thanks for that.
Yeah.
We've got a couple more general questions coming in, more kind of to do with VMS than the biology part. But so we've got a couple on YouTube. Shall we go with that one in the chat there? John Barry, and if you want to ask your question out loud, you're welcome to unmute yourself, John. Great. Okay, so John asks, what determines whether the fluids vent onto the seafloor or are replacement below the surface? Right,
yeah. I guess whether do they inhale or not? Yeah, I guess it's a question of the types of lithologies that are there. So the replacive deposits have been shown to be in, particularly we got pyroclastic material or on the edges of brecciated things like cryptodomes. They're very common in also in sedimented areas. So the Middle Valley deposit in off the coast of Wandafuka, because the fluids are coming up and they come into like a porous, effectively a sediment that's full of cold water, that acts like the seawater. It will quench the fluid and you actually form by replacement within effectively an aquifer. So whereas on mid-ocean ridges where it's basalts, they'll come straight up onto the seafloor. So my feeling is a lot of it's due to the rocks that are the footwall. So if you've got more brecciated and tufaceous material, much more opportunity for replacement. Obviously, brecciation is important. So tectonics and the facies, I think, was the biggest controls to those. And because if the fluid comes up and hits what it thinks is like the sea floor, because it's a cold aquifer full of basinal fluid or something, it will form within that unit, which is what's happening in Middle Valley.
Great, thanks for that. Jamie, do you want to ask that next one?
Yeah, sure. So this is from somebody in the chat. They said, basically, can you use the fossil record to prospect for VMS deposits? I think that's quite a good question, actually.
Yeah, no, it was. And it was one of the things why I got interested in the starting out, because I'll go back to the history of why I got interested in this. I worked in mineral exploration before I became an academic. So in the late 1980s, I was working in south of France doing mineral exploration. I mean, that's a fantastic thing in its own anyway, to do that. But we were working in this basin in the Cevennes region where there were some enormously large fossils developed that, and I talked to the local paleontologist and he said, well, the reason we think they're big is because there was hydrothermal venting and these organisms had basically were able to get more nutrients and got bigger. And so that was really why I started, because I thought, well, maybe these could be a prospecting tool because we could use them to look at where things were in the records. We suddenly found unusual organisms. And it's really true that this, if you look at the modern seafloor, 80% of the organisms around a vent are endemic to the vent. That means you don't only find them around vents. So that means if you found that organism, you'd say, must be a vent nearby. The only problem is, of course, we always find them at the vent. But it could be that we might find them, in other places where there's minor venting, and we get these unusual organisms in the fossil record, and you could then use them as a prospecting tool. So one of the things that we haven't done is that in some of these vent sites, you get benthic things like forams that like to go close to vent sites. Now, maybe we could find unusual forams. Now, these are more widely spread around vents, and perhaps they could be prospecting tools. We've not taken it to that stage, but I think there is, that's another good project to go on. And it's how I started. We've never got to that point of saying, can we now turn that into a prospecting tool? But I do think it could be.
Yeah, maybe at that kind of 100-meter scale or something like that for the forams. Yeah,
well, we certainly, those filaments in those the filaments I showed you in the jasper at Figueroa, we found this jasper unit that was full of bacterial filaments. That's a good example of something that you think, well, that's not a diagenetic chert that's just from, from sponge spicules or something. It could well be a hydrothermal chert. Now, the reason you think that is because it's got bacterial filaments in it, because they must have been living there on something. And so I would say that is a good prospecting tool. And we know that, we've done some work out in Løkken in Norway, and around the Løkken big massive sulfide deposits are these regional cherts, and they have bacterial filaments in. And effectively, that's the ore equivalent horizon. You can track it back to the massive sulfide. So in that case, the bacterial filaments can be used, at least as a regional tool.
Yeah, really interesting.
Yeah.
Cool. Okay, just if you are okay for another couple of minutes, we do have a couple of questions here.
A beer will call me soon, but
yeah, my dinner will call me soon too. Sandy, if you're there, you're very welcome to unmute and ask your question. Okay, I'll ask it. Great, thanks. Are there any areas or periods that you would specifically like to get specimens from? For example, the Arabian Numbian Shield?
Yeah, well, absolutely. I think I pointed on the. When I go back, maybe I can find the slide. We're particularly interested in that the Mesozoic to Cenozoic site, and then the end Permian. So things that were in the Permian, the Triassic. It's not very productive for VMS systems, sadly. But I mean, there are belts. So definitely looking for those geological transitions would be pretty important, I think.
I don't know if that was an offer from Sandy, if she's around the area and can get some.
Yeah, it'd be great. I mean, I'd say the thing to look for would be for some of those textures that I kind of mentioned that are, definitely indicative that you're on the seafloor and that there's good preservation. So, the presence of intercalated oxide sulfide sediments is a good one, because then you're close to these very, very fine pyritic layers, because they get very much overprinted by metamorphic events. So, and then obviously if you start finding these funny little annular rings of chalcopyrite and zoned minerals that look like they could be conduits or vent, because not only is that telling you you're in the right environment, they get really quickly overprinted. So that points to the fact that they're really nicely preserved. So that's what I would say to anybody who's out there looking. If you've got that kind of well-preserved, particularly fine-grained pyritic sulfides that could almost be microbialites, then that they would be really, really positive to look for. So yeah, would welcome the chance to work on any new stuff. So there, Nubian Shield sounds really good.
Great. Thanks, Sandy, for asking that question. Do get in touch with Richard if you have samples. Yeah. We do have a couple of kind of broader general questions on YouTube, but more around if you can give any insights on whether which VMS are particularly related to different metals. And I don't know, I could extend on that. Is there any of those particular life forms that are attracted in sites with different metals? I don't know if that's...
Yeah, I think we sort of talked a little bit about that earlier on. There is some evidence that perhaps some of the sites that are perhaps more zinc rich are lower temperature, therefore they maybe have their more oxidized fluids. They may have less reduced sulfur, so they may be less prospective. So there's a bit of circumstantial evidence. My colleague Valery Maslenikov has much stronger views on that. I just don't think we have enough samples yet to make that conclusion. That would be my, I always use Occam's razor, and I think I don't have enough evidence to say that's true.
That's fine. Maybe we'll have you back on next year or the year after. Maybe there'll be something.
Yeah, when we do find the link. Yeah.
Great. Jamie, do you have any other questions from your side?
So I was going to ask about the bacteria a little bit, because first things first, you've actually made fossils interesting for me. So there's a positive.
Look, as an ore deposit geologist, I feel the same, Jamie. Before I discovered these, I gave up paleontology in my second year, and I've now become fascinated by fossils, and it's probably a sad thing, but it's because these are so interesting.
Oh yeah, certainly. But I certainly haven't seen a talk which kind of shows that link between the bacteria and obviously the tube worms and the symbiotic relationship all that well. So I was just interested, how much do we really know about this bacteria, and how resilient is it to things like pH and temperature changes?
Good question. Well, one of these bacteria is used for the PCR reaction. So it's one of these thermophilic bacteria because some of these work at quite high elevated temperatures. They work really quickly. So you can get them to do reactions chop, and so they are really interesting, and quite a few of them have been patented, sadly. That's why there was such a race to make sure the human genome was released on open access, because you can imagine because you can patent genes. So these microorganisms get patented because they're very, very useful. They're tolerant of high temperatures, they're tolerant of chemistry, they're doing really weird reactions. So yeah, there's a heck of a lot of research, Jamie, going on into these vent bacteria, because they are so interesting, because they do weird reactions and they're very tolerant of what you would think would be quite toxic and aggressive environments.
Oh yeah, for sure. It certainly seems like they're almost the engine room, because they are the things that are reducing the sulfur, or they're certainly using it in their chemical reactions. And it's almost like if you remove that pin, the sort of system would collapse in on itself a little bit. So I was very like, what happens if, say, climate change starts affecting things at that level? You sort of pull the pin out, and these sort of vent systems, these communities start to, unfortunately die off. That was one of the things I was quite curious about.
Well, I guess we've got things in the Archean, but the VMS in the Archean could well have had bacteria there. So
yeah,
exactly, they may be a key part of maybe getting a system started. So yeah, I think, well, we find weathering, pyrite doesn't weather unless bacteria gets involved. So if you had pyrite in oxygen and water without any bacteria there, it would sit there quite happily, because of the the energy hump to get it to start oxidizing is too much. But the bugs get in there and they help it, and they start producing acid, and then everything dissolves, and you get acid mine drainage. Without bacteria, you wouldn't have AMD. So they also cause problems as well. But yeah, it's interesting as to how important they are. We know in the Irish oil field, without bacteria, you probably wouldn't have those big zinc deposits because bacterially reduced sulfur is absolutely essential for the formation of those deposits.
Oh yeah, it's critical. It seems critical for that. It seems critical for life, basically starting at these vent communities. I mean, that's a whole separate talk in itself, I imagine.
It is, yeah. A biologist for that one.
Yeah, for sure. No, really good. I mean, so I'm not really seeing anything else in the chat, Alannah, other than the fact that John's made a very good analogy, in the sense is that the organisms are living close to the edge are almost like the Sicilians living on the flanks of Mount Etna. I think that's a good analogy.
It is a good analogy. Humans will always push to living in, the delta in Bangladesh, because it's very fertile, but, you occasionally get flooded. So it's like that. I mean, a vent is like a delta, or you're right, or a volcano where the agriculture is good and you can grow really good grapes for wine.
Risk versus reward, I think, is
yeah.
Well, I think we're a little bit after 8 o'clock here for me, so we'll let you, Richard, have a beer and have some dinner. So thank you so much for joining us. Thank you everyone else for joining us this evening as well. And yeah, remember you can head over to the SEG traveling lecture session tomorrow. Do check that out on their website. And this is the final talk for us for the season. We'll be back in late September. So we wish you all a really great summer or winter if you're down under. And yeah, thanks so much, Richard, again. It was fantastic.
No, thank you very much. And yeah, have a good summer or winter, everybody. And yeah, looking forward to the next season, Alannah.